Senna. Its common name or synonym is Indian Senna, and biological source it consists of dried leaves and pods of Cassia angustifolia, that is Indian Senna or Tirunelveli Senna. And the other species is Cassia angustifolia. We call it as Alexandrian Senna. And its family is Legumiaceae. And geographical source, the Alexandrian Senna is indigenous to South Africa and it's also grown Egypt, Nile River and also cultivated in Sudan. The case of Indian Senna or Tirunelveli Senna is indigenous to southern Arabia and also cultivated Tirunelveli and Ramanathapuram district of Tamil Nadu. It grows Sindh and Punjab region. Next is the morphological characters of Senna. Its appearance generally entire and less broken in good condition, and its size 2 to 5 to 5 centimeter long and 7 to 8 or 7 to 9 wide, and its shape is lanceolate, and its apex acute with sharp spine, and margin is entire and flat, base is less asymmetrical, and the veins, these are reticulate anastomizing towards the margin, and the surface transverse and oblique impression less pubescent, that is hairy, and its texture flexible and less brittle, odor faint, color slight green, and taste bitter mucilage. Vein eyelet number 19 to 22.5 and stomatal index 14 to 20, and palisade ratio is 4 to 12. Next is the chemical constituents, mainly Senna containing glycosides, mainly androgen and diandronol glycosides. Leaf containing 3% glycosides and pods containing 1.5 to 2.5% glycosides. These are two types of androgen glycosides, that is homodiandronol glycosides and heterodiandronol glycosides. In the case of homodiandronol glycosides, Senoside A and Senoside B are the active principles. Senoside A 0.3 to 0.45% is present in Senna, and Senoside B 0.3 to 0.5% present in Senna. And next is heterodiandronol glycosides. These are Senoside C and Senoside D. The other glycoside, camphorol glycosides, are present in Senna. The other active principles, rennin, allonmodin, and flavonoids are present in Senna. Next is the chemical test, that is Brauntrager's test for anthraquinone glycosides. In this test, the leaves are treated with dilute HCl and boil and filter. To the filtrate, we are adding any organic solvents like benzene, ether, chloroform, then shake well. To this add ammonium solution. The ammoniacal layer become pink to red color, this indicating the presence of anthraquinone glycosides. Uses. Senna leaves are used as laxative, and Senna is having irritant action on large intestine, so it shows sleeping effect. That's why we are prescribed Senna along with carbonatives. And Senna is having stimulant cathartic action and also increases the tone of smooth muscles in large intestine. Next is allied drugs and substituent. First one is the Arabian Bombay and Mecca Senna. It's mainly wild Cassia angustifolia. That is the biological source. And its specifications, the leaflets are more elongated and darken in color. Also, the vein eyelet number is different. And next is the dog Senna, that is biological source is Cassia operata. And its specification, Leaves are broadly obovate with abruptly tapering apex. The venation is pinnate, and other specification is lower epidermis shows papillose cells.